Hello everyone, my name is Sumit. I am back with the new video. Today we will discuss about the DNA replication. DNA replication is the process of DNA replicate in a copies. That copies are the photocopies. The DNA makes multiple copies of itself. It is a biological polymerization which proceeds in the sequence of initiation, elongation and the termination. It is an enzyme catalyzed reaction. DNA polymerase is the main enzyme in the replication process. Following are the important steps involved in the DNA replication. We will discuss about one by one. In that, first one is initiation. In the initiation, DNA replication demands a high degree of accuracy because even a minute mistake would result in a mutations. This replication cannot initiate randomly at any point in a DNA. For the replication to begin, there is a particular region called the origin of the replication. This is the point where the replication originates. Replication begins with the spotting of the origin followed by the unwinding of the two DNA strands. Unzipping of a DNA strand in their entire length is not feasible due to high energy input. Hence, the first replication fork is created catalyzed by the helicase enzyme which unzips the DNA strands. Then next step is elongation. In the elongation, strands are separated. The polymerase enzyme start synthesizing the complementary sequence in each of the strands. The parental strands will act as a template for a newly synthesizing daughter strands. It is to be noted that elongation is unidirectional, that is DNA is always polymerized only in the 5' to 3' direction. Therefore, in one strand, the polymerized template 3' to 5', it is continuous, hence called continuous replication, while on the other strand, the template 5' to 3', it is a discontinuous replication. They occur as fragments called Okazaki fragments. The enzyme called DNA ligase joins them later. Then next step is termination. In the termination of replication occurs in a different ways in a different organism. In E. coli like organism, chromosomes are circular and this happen when the two replication forks between the two terminals meet each other. Then we will discuss about the role of enzyme in a DNA replication. DNA replication is a highly enzyme dependent process. There are many enzymes involved in the DNA replication which includes the enzyme DNA dependent, DNA polymerase, helicase, ligase, etc. Among them, DNA dependent DNA polymerase is the main enzyme. DNA replication process in prokaryotes. The DNA replication in prokaryotes take place by the two strands of DNA unwind at the origin of replication. Helicase open the DNA and replication forks are formed. Then the DNA is coated by the single strand binding proteins around the replication fork to prevent rewinding of the DNA. Then next, topoisomerase prevents the supercoiling of DNA. Then RNA primers are synthesized by the primase. These primers are complementary to the DNA strand. Then DNA polymerase 3 starts adding nucleotides at the end of the primers. This leading and lagging strand continue to elongate. Then the primers are removed and the gaps are filled with the DNA polymerase first and sealed by ligase. Then we will discuss about the DNA replication in eukaryotes. The DNA replication in eukaryotes is similar to the DNA replication in prokaryotes. However, the initiation process is more complex in eukaryotes than the prokaryotes. In eukaryotes, there are multiple origin of replication is present. A pre-replication complex is made with other initiator proteins. The process is entirely the same but the enzymes used are different. Example, in eukaryotes, the polymerization process is carried out by the enzyme full sigma, whereas in prokaryotes it is done by the DNA pole third. Thank you. This is all about the DNA replication.